Each year, Microsoft Research hosts hundreds of influential speakers from around the world, including leading scientists, renowned experts in technology, book authors, and leading academics, and makes videos of these lectures freely available.
Thank you for coming. My name is Amy Draves, and I'm here to introduce George Church, who's joining us as part of the Microsoft Research Visiting Speaker Series. George is here today to discuss his book, Regenesis: How Synthetic Biology Will Reinvent Nature and Ourselves. We are at a threshold of a new era of bioengineering. Imagine resurrecting extinct species or making humans immune to all viruses. A few years ago, this would have been science fiction, but today these scenarios are quickly becoming reality. It's a new world filled with possibilities and perhaps perils. George Church is a professor of genetics at the Harvard Medical School and the visionary behind the Personal Genome Project. His many honors include the Franklin Laureate for Achievement in Science. Please join me in giving him a very warm welcome.
Well, thank you, everyone. It's nice to be here. This is my conflict of interest slide. So these are some of the companies that have helped and other organizations that have helped us take some of our primitive ideas and develop them and get them so that they're out in the real world. Now, the big question that you should be asking of anybody like me and of this book is why? Why are we doing some of the things we're doing? Some of the things we're doing are quite exotic. I won't be able to talk about all these things, but you're certainly welcome to ask me during the questioning or afterwards, why should each of you do your personal genome? That's a big question. Why should we do genome engineering as opposed to genetic engineering? Why mirror life? Why should we reverse extinction for some key species? Why should we store 10 to the 24th bytes or more? And why should we study every neuron and talk to it? First, a little history. Bill and Paul and I did almost the same thing in 1968, which was we worked on a teletype that was hooked up to a GE 635 mainframe. Since then, we've kind of converged a little bit in our interests in terms of public health and biology and neuroscience, respectively. And both Paul and Bill have funded me, and I've gotten to know them. But the key thing, the point that I'm making here is about back then we borrowed mainframe computers. We time-shared. And eventually we made a decision to get our own. And I had borrowed many computers by the time this TRS Model 100 came out in 1983, and it of course had Microsoft inside, and it was listed at 1100, and when it came down to 380, my student wallet opened up, and I got it, and it's still running programs in my office, and whenever journalists come to my office and they see these computers are, running programs, that they get all misty-eyed about how they used to phone in at 300 baud their news stories on this thing. It was actually had quite an impact. It was 20 hours supposedly on AA batteries, and it had awesome 8K kilobytes of RAM. It's amazing what you could do with that much RAM. And no disk drive. So I'm going into the technicality of this because it represents how do we decide when we get our own genome, and how do we decide when we make a lot of technological leaps. So one of the deciding factors is cost. If cost comes down enough, even if there isn't a lot of application software, you will do it. And I should mention almost everything I'm talking about here is software one way or another. We might write it in hardware, we might write it in DNA, but so anyway, if you extrapolate, and extrapolation is always a dangerous business, and it's especially dangerous if you extrapolate exponentially. And optimistic exponential extrapolation was to say that DNA since the 60s was on the same curve as Moore's Law since the 60s. Well, very nice fit to both of them at a 1.5x exponential per year. So you could say, well, we did the first genome pretty badly for about $3 billion. When would it be accurate and available to all of us, affordable sort of in the low thousands? And the estimate was 50 plus years. You can see this extrapolates somewhere out here past 2040. Fortunately, we had an early arrival where the exponential changed. It's still an exponential, but it's a much steeper one. And I'm happy to say I was involved in that. And I don't know whether we should overinterpret the fact that it's flat now, but you could argue that if it's not coming down, it might be a good time to buy your genome now. It's in the low thousands. It's always hard to buy something when the price is exponentially decreasing. And I won't go through all the details of why this arrived in 6 years rather than 60 years, but I will give you one totally from the cutting edge of where it's going next, where it might start this exponential again, and I don't know at what slope. And that's called nanopores. And nanopores are basically a direct interface with single molecules where each single protein pore through which ions are going, sodiums or potassiums, and a very high resistance, sort of giga to teraohm resistance bilayer. And as a single polymer chain of DNA, single strand goes through there, it will selectively block the ionic conductance. And so you see here is picoamperes on the y-axis and time in milliseconds along here. And here little blocks of 3 Gs in a row and 3 Ts in a row. And you can see there's very nice discrimination between the 4 different kinds of bases, but there's not such great discrimination between a run of bases in a row. It's hard to tell whether that's 3 or more. So that you can get on the order of 4% error rate with these, and the good news is the instruments have come down in size from sort of mainframe size sequencing devices to handheld ones. This is not yet commercially available, but it is from a company, Oxford Nanopore. So you can either measure the polymers, meaning these long chains, or you could break it into monomers into A, Cs, Gs, and Ts and throw one at a time, and that might solve this problem. Or you can tag them. So you can actually put tags on each of the bases and monitor them. And that's probably the winning combination. This is the slowest of the new technologies to arrive. I've been working on this since the early 1990s. But here you can see how cleanly separated. This is a histogram, actually. It looks, you typically don't get tags that are anywhere close to the Cs and Gs and Ts. And so these are very well separated, baseline separated. And the error rate for detection of these things is on the order of, these are single molecule events with error detection around 5 times 10 to the 8th. That's now that's not a sequencing error rate, that's an analytic chemistry error rate, but it could turn into a raw single molecule sequencing error rate, which would be unprecedented and wonderful. Now, treating you all as both as geeky consumers, you want to know what kind of genome should you get. So you can get sort of, like in the sub $100, you can get SNP chip, single nucleotide polymorphism, but this is only 1/3000th of your genome, and it's not actually the best one. It's not even that informative fraction. The exome is specifically the parts of your genome which are most informative from a medical genetic standpoint. But it's only 1% of the genome, and there's all kinds of things it misses. Whole genome sounds pretty good. It's only 95%, but for some reason or other we all call it whole or complete genome. No one has ever finished actually a human genome or any major animal genome. But it misses something that a phased genome will give you. So if you're going to go out and buy it, which I think you should get a phased genome. And this means 2 hits in one gene can be distinguished from a hit in one gene and a hit in another. So each of you has 2 copies of every gene. And if you observe with these short reads that you have 2 mutations in that gene, you don't know, unless you phase it, whether it's just 2 hits in one gene copy and the other gene is fine and doing its job and making you fine. Or if you have a deleterious hit in both copies, then you have nothing from that gene, and that gene's important, and you're potentially got a time bomb going off. So get a phased genome is the take-home there. Second take-home is it's not that easy to go from your personal genome straight to traits, to determine the exact hour of your death and what you're going to die of. But there's all kinds of cool things you can do in between if you have a good cohort to help you interpret them. And we have this awesome cohort called at personalgenomes.org, and we collect all these data, all this intermediate and environmental data, in part because the cost of your inherited genome has come down 1,000,000-fold, so has the cost of collecting big data from of other kinds of omics. They're called genomics, immunomics, so forth. And these are environmental, so it's how your immune system responds to microorganisms, and how you develop your RNA and protein is all big data that we can collect now 1,000,000 times cheaper. This is now an international project. There are over 2,000 people in the Boston branch alone. The Canadian version can handle the rest of the world, and there are many other countries queuing up as well. We have an entrance exam. You have to get 100% on it. Now, this is, as it turns out, one of the best projects for establishing standards. And so the National Institute of Standards and Technology and the Food and Drug Administration have teamed up, which is great in teaming up to make a genome in a bottle, which means they're going to take a few genomes and make thousands of copies of them and send them out to every company that wants to develop new diagnostic tools, new instruments, anything that might involve genetics. And so when they look through all the different kinds of samples that were present in the world, they have quickly settled on ours as the main one, the only one that really properly has consented the people involved for re-identification and commercial use. Either one of those could be a showstopper. How is it that we have this unique cohort? And again, it's because everyone has to get 100% on an entrance exam. We educate first. This is the world's only open access data set, meaning we do not promise de-identification because we know that everyone can be re-identified. And probably people in this room understand better than anyone how in this world, this highly connected world, how easy it is from either your traits or your DNA, or both, in this case, you could identify people. We make stem cells available and many other cells available through a biobank so people can test the hypotheses coming out of these projects. And we're collecting even bigger data sets of all kinds of traits, not just highly medical traits, but almost anything that could impact our way of living, anything that can be measured. We're trying to measure it, and this includes skin resistivity, all sorts of cardiovascular components. This is a portable ultrasound. EEGs is not necessarily the best example. And then this is the one that isn't portable, which is fMRI, but we think it's a terrific stopgap for measuring the brain. This is actually slices through my brain. In fact, fortunately, they were virtual slices rather than real ones. Now, this, one of the reasons that people have hesitated in addition to price is they think, oh, I'm not going to learn anything about my genome. I'm just going to get this generic health advice, and it's not going to tell me anything. And if it did, there's nothing I could do about it. So I'm just going to, take this advice, exercise, drink milk, greens, grains, so forth. Well, it depends on your genetics. These aren't necessarily, if you exercise and you have hypertrophic cardiomyopathy, you can be dead. There are things you can do to handle it in advance, preventatively, if you know you have it, but it's not part of standard medical diagnosis. If you have lactose intolerance, that's not good for milk. Green beans, if you have favism with G6PD mutation, that can be a killer, and many drugs as well. Certain grains will set you off. You have celiac disease. Hemochromatosis is really bad if you have iron in your diet. And some people do take iron supplements not knowing they have hemochromatosis, and so forth. I'm a narcoleptic. So rest is not a problem. And that's just a short list. There are 2,700 of these that are highly predictive and medically actionable. These are not things that are weak effects. There are plenty of things that are weak effects. There are plenty of things we don't know anything about. But that's not what medicine is about. Medicine is about what works really well. And there are 2,700 that work really well. So here's audience participation. How many people here do not have any insurance whatsoever? Okay. And it can cost. This is kind of group insurance rates. I don't know how much, I won't ask you how much you're spending on insurance. But the fact is, if you get to the end of your life and you've been very, very healthy and you suddenly die and your house, nothing ever happened to it, never had auto accidents, you didn't need long-term care, didn't have any fires, anything, you could say you wasted $9,000 a year, right? But you didn't know that in advance. So the question is, why don't you have your genome sequenced, right? Because it could be it doesn't tell you anything, but you don't know that until you look at it, right? That's the end of my argument. And here's some examples of actionability. Not just actionability kind of in conventional medical genetics, but this is a new way of doing medical genetics where people are getting the whole genome sequence. They're not looking for a particular gene, which is kind of the way we've been doing it since the 80s. In this case, little Nick Volker, 3 years old, his guts were literally, fecal matter was leaking into his abdomen. He got dozens of intestinal surgeries. Clearly this was not going well. In desperation, they decided to sequence him. They found his gene here, IXAP, and they completely switched the therapy to a little cord blood transplant. He was fixed within weeks, and he is around the time that he's celebrating his 8th birthday. Beery twins misdiagnosed as cerebral palsy. They sequenced their whole genome, found this gene, and then they changed their diet to include serotonin and dopamine precursors, just a dietary change. Dr. Wortman actually studied leukemia and ironically got leukemia, no connection, just coincidence, and then his genetics told him what drugs would be effective. Gilbert was an example of many people who get either mastectomies or ovarectomies if they have BRCA1 or 2 alleles. It's a preventative thing, but it's increasingly applied. And here's a case, a whole family had not breast cancer predisposition, but stomach cancer. This was killing their family off at early age. And 10 out of 18 people that were tested in this family had it and had their stomach surgery preventatively. So there are things that you can do, some of them relatively simple, from a whole genome sequence. You don't necessarily need to know exactly what gene you're looking at. And this raises an interesting new way of doing science as well as doing medicine, which is we've been building up these gigantic cohorts where to even draw a single weak weak correlation, we have to have 10,000 people. But what if we have one person? Can we do anything that's maybe even more powerful than correlation, which has to do with causality? And furthermore, I'm going to take this in another strange direction, which is, are we just talking about rare deleterious alleles? Are there rare protective alleles that give us superpowers or interesting properties, or maybe mixture of good and bad? And here's an example where a double null, meaning knocking out both copies of the myostatin gene, results in high muscle growth and decreased body fat. Flex Wheeler does work out, but he obviously has an advantage, and the causality here can be proven in animal models. Here's 3 different animal species, mouse, dog, and cow, that all have the same effect when they have both copies of their gene knocked out. So that's myostatin. But there are many other ones, and this is just a partial list. LRP5 produces bones that are so strong that it'll break surgical drill bits. PCSK9 reduces your coronary artery disease by about 90%, and is an inspiration for a new antibody-based drug. CCR5 is an inspiration for a drug we'll see in the next slide. FUT2 and CCR5 both result in virus resistance. FUT2, double null, results in norovirus resistance. And then here's my favorite, APP gives you decades of extra life without Alzheimer's. In addition, ApoE4 is an Alzheimer's risk. It's about 20 times higher risk than if you have 2 copies of ApoE3. I fortunately have very low Alzheimer's risk, as I know that. That was of significance to my family because my father had problems. So more of this n equals 1. This n equals 1 patient he happened to be one of the most unlucky people on the planet. He had both leukemia and AIDS. They treated his leukemia in a fairly conventional way, which was to find a stem cell donor who was matched for his HLA, so it wouldn't reject the graft. But they happened to also luck out they could find a donor who was not only matched for not rejecting, but it had a double null, both copies knocked out of the CCR5 gene, which is how the AIDS virus gets into the cell. It binds to the CCR5. So if you knock out both copies, there's no CCR5 on the T cells. And he got cured of both diseases very quickly. And 5 years later, he's still disease-free, as far as I know, from both diseases. And inspired a clinical trial which went through phase 1 and now phase 2, which means it's low toxicity and efficacious, where you take the cells out of a person, treat their own cells with a molecular machine, a zinc finger nuclease it's called, which will cleave specifically just in the CCR5 gene. So it's not putting some random package anywhere around the genome where it can cause trouble. It's specifically making a double null in CCR5. You put it back in the patient, and now they have a bunch of T cells that are resistant to the AIDS virus. This is just what those zinc fingers look like. They're called fingers because they have these alpha helices. This is a sort of nanometer scale. Here's a little zinc atom that holds it in place next to this DNA sequence. So you have a very specific code where you can tell how this protein will recognize the DNA. And then you have a similar code now for a newer class of proteins called tau, or talins. As I mentioned, we're doing big data on the various omes, including microbiomes and immunomes. We can not only detect antibiotic resistance, but there are consequences of antibiotic treatment where you can, if you knock out enough of your microorganisms, the one will stay behind, which is Clostridium difficile, and this will cause all kinds of gastrointestinal problems. One of the solutions, which is actually pretty low tech, is called fecal transplant. So you can actually change your microbiome, and it's probably affecting obesity and diabetes as well as and you can change your immune system as well. We go around the world and you can find people that are resistant to HIV. They're very rare. I mentioned one already, that one way that people can rarely be resistant is removing the receptor. But the other way is they can actually make an antibody that's neutralizing. It's not easy to do this with a vaccine, but you can do it with, you can find a rare person who happened to hit the jackpot, not because they had just the right vaccine, the right virus, but because they had luck with their immune system. And you can take that and put it into other people. So far, this is at the rodent preclinical testing stage. Another way we can deliver antibodies, aside from having your muscle cells make antibodies, which is what's happening with those HIV neutralizing antibodies, is we can make these nano robots, which are a little bit smarter than an antibody, that they contain antibodies. These little gray things are antibodies. They're color-coded purple here. And all the rod-shaped things are DNA double helices. You can design this basket, basically, which is held together when these two come together in long rods. That's just 2 points that are holding this basket together. And when it opens up, it delivers this payload, which can either kill specific cancers without killing any other cells, or it can do immune modulation. And it only opens up when both of these double helices breathe enough so that one half of them will bind to some protein on the surface of a cancer cell and recognize it. And when both A and B do that, they no longer can bind to their partners, and so that there's nothing holding this hinge thing closed anymore, and it opens up and does its payload. So this satisfies some of the main criteria for what we would call a robot. It has sensors, which are these aptamers here that make a transition. It has logic, so it's A and B, and you can have more complicated Boolean logic. And it has actuators, which it opens up and then delivers a payload, which can be toxic or modulating. We're going to come back to nanorobots and DNA encoding in just a moment. The biobank that I mentioned that we have for stem cells in the Personal Genome Project can also be used on organs on chips. So this is getting pretty sophisticated, where you can test drugs for their toxicity in a human rather than on human cells rather than animal cells, but in a much more realistic way. So, for example, here's a heart and that has contractility, and a lung that has air, epithelium, endothelium, and blood, and it flexes with the air going in and out. So all these mechanical forces are much more realistic. And there's 10 of these, including a neural and blood-brain barrier, which brings us to our last topic, which is brain activity map. We want people to be there's a whole variety of neurodegenerative diseases, traumatic injuries. This particular woman is tetraplegic, can't control arms or legs. Parkinson's, Alzheimer's, many diseases that have a major neural component. And we'd like to be able to input and output to them to digital computers. In this case, she has a small number of electrodes going into her brain at this point. And for the first time in her life, she's been able to give herself a drink here by manipulating through her brain waves this robotic arm. Now, this is some of the work from my close colleague, John Donahue. But the point is that this is still very crude and slow, and by looking only at a few neurons, it's amazing that we can do anything what we could do with a larger number of neurons. One of the problems with cramming more electronics into a brain, even if they're just wires, is that the efficiency of integrated circuits in terms of the amount of energy that they need to operate, if you wanted to make this, say, wireless or in a variety of senses, either record or do input and output, there's been this Moore's law, not just for density of circuitry, but also for energy efficiency. And you can see that we're off by about 10 orders of magnitude from where biological systems can do operations at about almost 10 to the 26 operations per kilowatt hour. An example of one of these that we're harnessing is DNA polymerase. This can go up to about 1 kilohertz, depending on the polymerase. And these polymerases we've shown, and others have shown, that they're responsive to ions. Remember I showed you earlier a nanopore where you can convert DNA into modulation of ion channels. Here you can take ion channels and use it to modulate DNA synthesis, so we can actually get misincorporation of bases, making a ticker tape essentially, or a long digital recording where DNA is the digital media. So this is very compact. It's about 1 billion times more compact than any other digital media, and it has very low energy components, as I mentioned just a moment. So here's a paper we just published in Science where we did another kind of digital encoding. So that was one intended for monitoring neurons. This one was purely digital, where we did an inkjet using organic chemistry to encode a book, 5.2 megabits, which were 5.2 mega base pairs of DNA. And and this kind of exceeded the density in terms of bits per cubic millimeter of most other technologies. And the next step that we'd like to do is we're developing, this was digital to digital, essentially digital zeros and ones to ACs, Gs, and Ts. And next we're going analog to digital, so taking photons and audio-visual data and converting it directly to DNA with no mechanical or electronic components, purely photons to DNA sequence. Here's Stephen holding a book that you may recognize, and holding 20 million copies of that book in DNA form, which is actually a tiny dot in the middle of this red ring here. If you missed this episode, he immediately wanted to eat it, and I stopped him. Not that it's that particularly valuable. 20 million copies here or there doesn't really matter. We made 70 billion of them. So I covered some of these topics here, ending a tiny bit early here. Hopefully I've stimulated some questions. I think I gave you a few reasons why I think personal genomes is now a very low cost. It's like insurance. You should get it whether you are going to need it or not. Genome engineering, I barely got to touch on, but we've used this for engineering chemicals and fuel now being produced at about $1.28 a gallon. Mirror life, that would be very hard to explain, as well as de-extinction. Yottabytes, I think the audiovisual analysis goal that we have, you can imagine having ubiquitous audiovisual recording where you would be basically archival storage, where you wouldn't read it unless something interesting, exotic, or really good or really bad happened, and then you would go and you would selectively read just a few gigapixels from that would be relevant. Period, full stop. Thank you. Question.
So I've actually been reading your book. It's a great book. However, and I was a little discouraged by your last comment, I was a little bit lost about mirror life. And so is there any way you can give like a 30-second layman's term as to what that is? I was lost on that.
Okay, yeah. It's really counterintuitive. So let's see. Molecules in the world can have handedness. Not all of them do, but some of them do. So like your left and your right hand, when you hold it up to a mirror, you get a mirror image of it, and but you can't rotate it. So I can't rotate my right hand and make it fit on top of my left. Right. The only way I can make it look like my left is through a mirror. So that's true with molecules too. And almost every molecule in your body, not all, but almost all, is chiral. It has a handedness to it. And
now, does the handedness literally mean the physical layout of the molecule? Like it'll
right. So think of a tetrahedron, right? Where each of the balls on the vertices of the tetrahedron are labeled in different. That's what the molecules are like. And it's really because carbon has tetrahedral symmetry and has this, if everything were made up of simpler bonds, then we wouldn't have this problem.
So the idea is to make a form of life which we can keep under control because it's completely separate from our ecosystem?
Well, the separate from our ecosystem is partially true, and the keeping under control is only if we intentionally do that. Because here's a scenario, which is if you if we were to create a mirror life, it wouldn't be completely separate from our ecosystem in that there are a few foods that are common. So acetate, glycerol, and glycine are three top ones. And so they could steal all of that and incorporate it into their body. They could not be eaten by anything alive today, right? Because all of the enzymes, all the viruses and everything have a certain handedness they expect. So so you'd have to be very intentional about this. And in fact, many of the things I'm raising in the book are not because I'm into science fiction or anything. They're actually approaching much more quickly than we think. And we need to talk about them a little bit in advance so that we don't just accidentally create something that will suck down all the carbon dioxide.
Algae for creating biofuel. One of the problems with them is they get sick.
They do.
Presumably this would create a form of, if you could do that for all algae, it wouldn't be infected by anything existing.
That's exactly right. But you need to be very careful. You need to have it on a leash. So algae are particularly hazardous because unlike, say, E. coli, which is another industrial microorganism that we work with a lot, that one actually has to eat food. But algae is just using minerals and carbon dioxide, all the inorganic stuff. And so literally it could suck down all the carbon dioxide in the atmosphere. So we may think we have too much carbon dioxide right now, but if we lost it all, we'd have an opposite problem to global warming. So I'm not sure I answered your question completely, but.
So the significance, like if you had a molecule and you create a mirror of it,
right.
What's the...
So you do that chemically.
Yeah.
You could do that chemically.
That's what purpose, why?
So why? Okay, so here is one description. If we can keep them under control, we can increase productivity, and done right, we can increase safety. Imagine that everything in the world that rots, rope, clothing, wood, so forth, these are actually good materials, but they're displaced by materials to the extent that they can be displaced that don't rot. But you can make biological materials that would persist in the human body without causing the immune response, or the much lower immune response, and they won't degrade. So artificial, molecular devices I described, or even physically large devices, implants and so forth. It also brings a whole new set of chemistry. It's not just twice as much chemistry, right, in left-handed. It's all that at every position in the polymer, you can think of it as a long tape. It can be either right or left-handed. So it's not just 2, it's 2 to the n, where n is the length of your polymer.
So in the science fiction book by Peter Watts, Starfish, it's about mirror-like organisms that just happen to have a faster metabolism, so they eat us out of house and home.
They don't even have to have faster metabolism. All they have to do is not die. Yeah.
So I was going to ask what the benefits, other than a doomsday scenario, would be for studying mirror-like, and I think you answered in terms of these longer things, which I thought about. Would that then imply that you would create a mirror-life ability to break that down, so that otherwise we just have like superplastics that don't even break down?
Well, I think we have to be careful about that. I mean, we have to be careful not only about the things we create, but the things we use to control them. Because let's take antibiotics. Okay, there's quite a large number of them, and one by one we're running out of them, right? Maybe had 12 major classes, 100 minor classes, and as they develop drug resistance, they're no longer useful. You just cross off the list. We only have 2 kinds of chirality, right and left-handed. If we use up the second one, that's it. Okay, so that's one way to do it. The other way is to put them on a metabolic leash. That is to say, they are dependent upon something that only we have in the laboratory, right? And that's even easier to do for mirror life, but it's something there are ways we can make virus resistant without mirror, so we change the genetic code. It's another way of making virus resistant. But all of these things we developed, we're practicing under controlled conditions how we can keep them on a leash, and really testing that, not just assuming because it looks good on paper it's going to work. So we have physical isolation, genetic isolation, every kind of isolation we can, so we can do the tests on a small scale first.
So last decade was gray goo, and this decade's like ambidextrous pink goo. Is kind of,
yeah.
All right. Okay.
Yeah, and gray goo too. Yeah.
One other question. I read recently a subject and wanted to see if you could comment or were aware of it, that there's some discovery that there was an RNA marker in any cell that was infected with a virus that they could use to detect that cell has a virus and then cause it to self-destruct. Have you heard of this?
Yeah, well,
it's pretty remarkable.
Yeah, we heard of it and we use it as a tool. So almost so a lot of biologists, whenever they see a new tool, they apply it to their biology. Whenever we see new biology, we apply it as a tool. Okay. And I think what you're referring to is the CRISPR system, but there are a few like that. Basically, there's two major kinds of immune response. One is the one I described, which are protein antibodies that float throughout your blood system, and the other one are kind of intracellular, which are RNA that you get an immune interferon response, sometimes called. So they have all kinds of way of recognizing something's wrong with their nucleic acids inside the cell. And these are great tools. You can use them not only for diagnosis and fighting viruses, as you mentioned, but you can also use them for engineering genomes, which is one of my favorite things. So
my question was, are there good viruses, or is this, like, it seemed like it's pretty remarkable, like any virus they could get rid of out of your system. Like, is there a downside to that?
So there are definitely good bacteria, right? So I mentioned the case where if you don't have a good set of bacteria, Clostridia difficile will take over your gastrointestinal, and that you do not want. There are very few examples of good viruses. Probably we're ignorant, but we're not that ignorant. They're satellite viruses that kind of keep the other viruses in check. There are a few. There are not as many good examples of good viruses as there are bacteria.
Is this as big a panacea as it sounds? I mean, I read that and go like, holy cow, that's...
Well, so the tests that we have done is, we meaning the field, is you can grow mice in completely germ-free conditions, multiple generations, and they're okay. If anything, they're missing a few bacteria that help the vascularization of their gastrointestinal system. So there is some developmental feedback there, but there's no known consequence of having no viruses. Now we have viruses in our own genomes. So these are endogenous retroviruses and other things like that. And they go wacky in your brain. So during brain development, you get a lot of these things jumping around. And it's still an open question as to whether these are good for your brain, bad for your brain, or neutral. And so the only way to test them is using genome engineering to get rid of them from, say, the mouse genome. And this is very significant in the sense that when these things, if we're recreating extinct species or doing transplants between pigs and humans, there's one of the concerns is that one of these retroviruses will jump species because of this new intimate relationship. So,
A couple of online questions.
Yeah, sure.
There's a lot of interest in the personal genomics. So, question about how do you get the phased genomics?
Yes. Right. So we published this in Nature just this July. I may have the reference here at the bottom. Yeah, no, I don't. But anyway, it was in Nature in July in collaboration with Complete Genomics. The way it's done is really cute. You take as few as 10 cells, 5 to 10 cells, you break them up so their DNA gets a little bit broken, and then you spread it out into a large number of small wells. So it's 384, but it could be anything around that. And in each well is about 1/10 of a genome equivalent. And then you amplify that up with an enzyme, and now in that well is that fraction of the genome. And now the chances are that you didn't get 2 copies of each piece of DNA, the one from your mother and one from your father. So you actually get these long things called haplotypes where you get basically, that all the DNA came from a piece of one of the 2 chromosomes. And so you don't get confused between the 2. And that eliminates all kinds of errors, and it establishes phase that that these all came from your father's chromosome or your mother's chromosome. And so you get 2 for 1. It's about 1 error in 10 million. I forgot to mention this. So you get both about 100 times better accuracy, and you get phasing. Yeah, so look in July in Nature.
There was another question about what you think about the privacy aspect. If it's one thing to do it for yourself, but when insurance companies start to get involved.
Right. So privacy has changed a little bit since we started this project in 2005. In 2008, Congress passed a law called the Genetic Information Non-Discrimination Act, or GINA. And that prevents employers and health insurance from discriminating based on genetics, no matter how they get it. Whether they get it from your sister or whether they get it from you or whatever, they can't use it. And I think that's got teeth in it, and I can't imagine why they would mess around with it. It doesn't affect long-term care and life insurance, but I think right now, even though there's no law, no insurance company that I know of is doing anything to discriminate based on that. So probably what's happening is the opposite, which is people like me that know that they're ApoE3 homozygotes and are probably not going to get Alzheimer's. Why do I have any long-term care, right? And so what'll happen is most of the people that'll stay in that game will be people that are very high risk of getting Alzheimer's and other things that require early long-term care. So I think it's going to work the other way around. Privacy, I think it's one of these questions. It's a kind of open question. Is our genome going to be like our face? Because our face right now is we have kind of a contract with society that we're going to share our face, and our face tells you all kinds of things about us that you might not want to share. It tells us pretty accurately your age, how healthy you are, what kind of emotional state you're in, your ancestry, just all kinds of stuff, right? And if you covered your face, you could keep that stuff private. But that's not really acceptable social behavior for most of us. And it may or may not be the case for genomes. It's like we are in a historical moment where we're deciding whether it's going to be like faces or whether it's going to be like something super secret. Yeah.
Sorry, I got a ton of questions. Could you describe the process of actually, I'm 40-year-old male. Like, if I wanted to change a gene, what is the process today of changing that? And is it just you change a little bit of the gene, like a segment of the gene in a few cells, or is it your entire body, and does it stay locked in?
Right. So, gene therapy was very bad 10 years ago. You would take a virus that would deliver a new gene that you were missing, and it would deliver it all over your body, and it would misdeliver it quite frequently, and it would cause cancer. So instead of curing a disease, it causes a disease. So that wasn't good, and so that set the field back 10 years, more or less. Now we've got this new generation of technology that's really awesome. You have the zinc finger nucleases, or the talons that will go to a specific place, make a very specific, you can think of it kind of like a little nano robot. It goes to the right place and it makes a fix to one, it can make it to one gene or both copies of that gene if you have to knock out both copies. Efficiency needs to be improved.
So question, so is it take along with it that section of the base pairs and cut and insert, or what?
It can. So that particular clinical study that I described from Sangamo, which is a little company I had a small role in starting in the 90s, in that case, they're just trying to screw up the gene. So it makes a double strand break, and it lets the body kind of, or the cell, fix it with an error-prone way.
So it's essentially just kind of cutting it out.
But there are plenty, we have the technology that we use all the time in the lab for bringing in a piece of DNA and patching it in perfectly. So you can actually replace a piece of DNA with another piece of DNA. It just hasn't made it into clinical trials yet, but it's certainly, we do it on human cells. And in fact, I have a center that I run that's entirely dedicated to human genome engineering of that sort.
And does it lock in? I mean, once you've changed it,
it's locked. It's not temporary. Well, it's not all your cells. So getting all your cells requires efficiency and delivery that we don't currently have. But we have pretty good efficiency, and we're getting better at delivery. Right now, if you want to make sure you've got it, you take the cells out of your body, you change maybe half of them, then you get rid of the half that weren't changed, and you put those half back where they belong, and you have to help it out a little bit. But I fully expect that as fast as this technology is going, we will be able to get any tissue to any degree of replacement. Yeah.
Is gene worded in such a way, the genetic information, non-discrimination, is it worded in a way that it's non-discriminative against the genes that you get naturally versus inverse discrimination if this technology continues to take off, right, and we start adding artificially created genes for competitive advantage inside, does the law discriminate against inverse discrimination?
I think that law does not address that question at all. I mean, yeah.
So when they say genes, they mean the ones you're born with, as opposed to 10 years from now.
That is what they mean. But I think this was just like a first step. I think there's going to be a flurry of legal activity in the future on this, both positive and negative. But so far, it was almost addressing a non-issue. There were really no examples of genetic discrimination either in health insurance or employment. The closest you came is occasionally a basketball team would require somebody to get a test for hypertrophic cardiomyopathy, which is actually for their own good, but that was considered discrimination.
I think we'll leave it with that. Thank you so much for coming.